So hello everyone, I'm Damien Thompson from the Department of Physics and Barnell Institute in UL. I'm leading the modelling strand of SSPC, Pharm 5, along with Harry Vandenacker. And the part that I'm leading is the atomic scale design. So Harry does stuff on fluid mechanics, or fluid dynamics, which is at the macro scale. We're down at the nanoscale, the level of molecules. So the title of this webinar is Molecular Design of Next Generation APIs. So the idea is that you use these big supercomputers, like shown here. We have a very big one in Ireland, which we make good use of. We also make use of high-performance computing platforms across Europe, and we use these to try to design big molecules. So more complex APIs, getting away from traditional small molecule drugs, say tetracyclines and so on, and moving towards large organic materials. So these are biomolecules. They're wet, they're soft, they're floppy. They have fantastic potential in terms of functionality if we can figure out how to actually deliver them and target particular sites in the body. It could provide drugs, but they're also very complex to work with. We have to learn a lot about the interactions that they have with each other and also with surfaces, so biological surfaces and also surfaces, say, of just, say, storage vessels. If you have a suspension of proteins in beaker, how do you stop them sticking to the surface of the beaker? So there's a lot to understand about proteins. Also stopping them sticking together and aggregating and forming plaques is obviously a big no-no for the use of these types of large molecules in pharma. But nevertheless, if we can overcome those types of existing bottlenecks, then there's fantastic opportunity to use those to make better, more effective drugs and replace the current largely insoluble small molecules that we have in pharma. So that's the idea, that's the motivation for this work. So hopefully I won't keep you too long. I'll start with introduction to atomic scale modeling in Pharm 5. So that's not just me, also Anthony Riley in DCU is heavily involved in this, and David Tiana in UCC is also involved, along with a lot of postdocs and PhD students. So it's a large effort within SSPC Pharm 5. Sorry. I'll then go on and describe design rules for predictive atomic scale modeling. So why is it difficult? Why do we have to remember to forget everything we think we know about how things work as large creatures in the at the scale of seconds and meters? Doesn't really help us very much to understand how molecules work. So there's some new physics that we have to learn to design molecules and design supermolecular complexes. And yeah, computers can be enormously helpful because they can go where experiments can't, and they can also speed up experiments by predicting the behavior of these complex materials. And before you ever do experiments, they can save money. If they work properly, you can save a lot of money. I'll then briefly describe some recent work we've been involved with, for example, controlling protein immobilization on surfaces, also trying to engineer protein-protein complexes, and also complexes between RNA and cyclodextrins, which were used for drug delivery. And then at the end, I'd say a little bit about the challenges in designing peptide nanostructures. So it is difficult, and we do need a lot of computing power to tackle it. So within Team 5 of SSPC, we're addressing key challenges by developing new techniques to design and predict. So we are predictive design behavior in silico in order to reduce trial and error experimentation. So that's how we're going beyond the state of the art, because we're becoming predictive. Instead of just using modeling to make pretty pictures to rationalize experiments, which is what a lot of modeling actually involves, a lot of design, we're going predictive. So for that, we need to really smarten up how we do it. We need to develop new codes. We need to get better at benchmarking against experimental data. So that's why it's very useful for us to be part of the large SSPC infrastructure. So I'll present on these first two aspects, focusing on molecular and nanoscale interactions. And within SSPC, within this modeling strand, we also have our mathematician colleagues here at UL, and Harry Vandenacker's group, also at UL, for people who are interested in things like simulation of industrial crystallizers and so on. So for this webinar, we are at the molecular level, but we do, as a group, we do a lot of other types of modeling as well. So yeah, we have developing a framework for understanding and predicting multi-component solid forms from first principles. Also then deciphering the nanoscale design rules of complex API formulations. So we're going from crystals to assemblies and trying to understand how they stick together. So what do? Well, we employ first principles modeling. So that's, you may have heard of the expression DFT, so that's density functional theory. It's a flavor of quantum mechanics that allows you to actually calculate the properties of crystals. So you can calculate the stabilization of crystals, you can calculate, say, the stability of co-crystals, and you can even, with some very sophisticated methods which we're developing, predict the solubility of crystals. So by doing this, then we can improve rapid informatics-based approaches to predict likely multi-component solid forms and their properties. So not only do we have to develop better codes, we have to develop faster codes. So there is that trade-off between accuracy and time to solution, which we're very, very aware of. So the picture here shows computed molecular dipole and the corresponding electron density on the right in a molecular crystal. These are the types of things we can routinely generate. Sometimes the calculations take a long time, so we are working hard to speed them up as well. So these types of simulations, well, they can identify non-intuitive crystal phases and polymorphs that can have novel properties. So you can find properties, you can predict properties that you would never really stumble across just from doing experiments or from pen and paper. So this specific example shown here is of an expectedly high piezoelectricity in the amino acid crystal beta glycine. That we discovered that using first principles models and then measured it experimentally. So we'd never have found it if we'd been able to do the experiments beforehand. So building on our expertise in high performance computing as a group, and also quantum mechanical calculations, and combining with machine learning algorithms, which we're starting to learn about, we can push beyond the state of the art. So basically trying to develop new transformative physical models that are sufficiently fast, accurate, and intuitive. So they have to be all those three things. There's no point being accurate unless it's fast. If it takes forever to get the answer, it's not good to anybody. And intuitive, so that can be used as a black box to some extent by, for example, say, industry collaborators. For broad uptake in drug product manufacturers. Not everyone needs to be an expert in physics to use these codes. That's what we want to get them out into the world. So we really would like broad uptake in drug product manufacture. That would be a fantastic outcome from SSPC if we actually achieve that. So showing here, for example, a map for modeling specifically co-crystals, I want to make the point that they're all interconnected. So you have to work on all these things at the same time. That's why it takes a lot of time and effort to actually get this right. So this idea of computationally guided materials design, there's a lot of moving parts. It's a many-body problem. So before we can truly pick a copomer or crystal component, we need to understand how it will affect structure and properties and how we can control this. So methods exist, I would say limited methods exist for each bit, but our challenge now is to put them together and advance them together. So I'll try to explain how we propose to develop modeling tools that will be truly predictive for things like, say, solubility, tabletability, things which are very, very relevant for industry. So Anthony Riley's work up in DCU is super important for SSPC modeling because he's developing really sophisticated methods to calculate dispersion interactions. So he's shown, very beautiful work, that these dispersion or these van der Waals interactions quickly become the dominant interaction in molecular crystals as the molecule size grows. So there's really no point looking at molecules in isolation, and there's really no point in looking at molecules in crystals unless you can treat van der Waals interactions. Because they're large, because there's large area contacts in the crystals, these individually weak interactions become very important. So they are glue that holds the crystal together with obviously electrostatic interactions. So this many-body nature is central to understanding polymorphism and also the properties in things like, say, aspirin and also even glycine. So glycine, I mean, if you think about it, it's the smallest amino acid. It's all hydrogen bonding that keeps it together, you would think. But if you don't actually accurately calculate the van der Waals interactions in there, you don't get the right answers. So many studies and approaches to co-crystals only focus on pairwise interactions. So for example, you build a model of a dimer, let it spin around in solution. And if you do that, you ignore the real complex picture in the solid state. If you like, you ignore the supramolecular dipoles in the crystal. So you really do not get the right answer. So what we do here is we look at the crystals and then we kind of do a dissect and rebuild approach. So we compare fragments to full crystal structures using faster methods for calculating dispersion interactions and also matched molecular pair analysis. So that's a method where we can explore very quickly and in a surprisingly powerful way actually, in a surprisingly predictive way, how changing single functional groups affect binding. So for all of this, we do need a lot of validation. So we need that close synergy with experimental works with people like, say, David Tiana in UCC doing lots of very nice work, including, for example, developing faster methods and developing force fields would allow us to reduce the time to solution. But always we need to benchmark, so we need to sanity check everything we do. So we need that close synergy with experimental work. So that's why it's very important for us to be part of the SSPC community and to refine our models that we develop through careful comparison and experiment. And then once we're benchmarked, we're in business. We can then become truly predictive. And once we have that, once we have sufficiently benchmarked them and sanity checked them, then we go predictive. Then we actually guide experiment. Some of the work we do here also in terms of crystal design is the predicting elastic properties. So that's, for example, that's very important. For example, if you want to understand and predict tabletability. So what Anthony's work showed here is that both many-body effects and also thermal contributions are very, very important in probability prediction. So what's shown here is calculations that he did on the Young's modulus, the stress-strain response of ammonia, but with and without van der Waals corrections and with and without thermal corrections comparing with experiments. So you need all those weak van der Waals interactions. You need those, in some cases, quite expensive and quite complicated many-body corrections to get the right answer. So you do need to throw money at the problem. You need your high-performance computing, and yeah, you don't get away with using cheaper models. Nevertheless, once we actually have models that work, then we can think about how we compare them back. But we do need to start with something that directly matches experiment, and we've got that now in terms of elastic properties. So we're developing workflows for using first principles calculations in tandem with quasi-harmonic calculations to make finite temperature prediction of elastic constants and to make that routine. So to try and speed up the workflow and where we can pare back the computation complexity. And again, we need to speak with you guys. So we need experimental validation. We need to use things like nanoindentation to actually make sense of the results that we get. So we're starting from things like ammonia, like aspirin, that are very well benchmarked. So experimental results exist for those. But as we go towards more complex crystals, more complex APIs, we need the live collaboration with experiments in terms of actually sanity checking our methods. And the hope then is that we can actually develop predictive models of tabletability from that. So yeah, we don't rely solely on quantum mechanics. We also then try to develop more high throughput screens. Once we have the good match with experiment, we can think about how to speed up the calculation. So use molecular dynamics, so essentially classical mechanics methods, and also other types of multi-scale modeling. Okay, so in terms of trying to understand then how to use large molecules, macromolecules, biomolecules, understand how they assemble together. The idea is that we scale up to these large fibrillar crystals by parameterizing classical models. So we need to go away from purely mechanical models because they won't scale to thousands and millions of atoms as required for this, for looking at things like peptides, the more complex APIs. So we use these classical models that can handle millions of atoms and microseconds and longer timescales. So we get closer to experiment. That requires further development of force fields, obviously. Then also methods like accelerated sampling and non-equilibrium molecular dynamics methods, which are being developed by some very clever postdocs in my group. And that's all facilitated by access to the Irish supercomputer. So the Irish Centre for High-End Computing has been very good to us. It's supported by SSI also. And we get access to close enough to petascale computing there. So we can run our calculations on highly integrated, highly parallelized supercomputers. So we can run very large calculations and get the answer quickly. So that allows us to stay competitive internationally. In this field, we're obviously competing against other groups in the world, and how big your computer is does matter. We've got a big one, so it's up to us to make good use of it. And certainly the work in SSPC is a very good use of the national supercomputer facilities. So what do we do? What do we use it for? Well, we look at things like mapping the assembly of intrinsically disordered peptides into insoluble plaques. So here the challenge is that you can never really sample for long enough, because unlike globular proteins, these short peptides, they're shapeshifters. They form millions of different poses in solution. So trying to capture all those is a huge challenge for simulation. But it's required because you can't do it. You can't capture them experimentally. They're too disordered. Each conformation is too short-lived, and only a vanishingly small subset of possible poses is actually implicated in activity. So only a very few of those poses actually make it into plaques, and it's up to the simulations to try and identify those and figure out how they do it. How do they stick together? What kind of geometries do they form? So that's one project we've been involved in. Also things like, say, stabilization of antibodies by excipients in solution. So there we did very large calculations to look at how immunoglobulin antibodies, how their shape and their activity can be preserved in solution. So if you put them in water, they tend to denature over time. What we found is, if you add a small amount of, say, amino acids or sugar to the solution, you can actually protect the secondary structure of the protein for longer. So you keep its active form for longer by using these excipients. So again, we can scan a large range of different excipients in solution using our computer simulation, so we save a lot of experimental time. So we can hone in on the most promising candidates before actually testing them experimentally. And we're also involved in predicting the most favorable packing orientations of non-natural amino acid crystals. So how do you develop new types of peptides which have properties when they assemble together which were not necessarily selected for by evolution, which we could choose in our industry applications. So we can summarize that in these types of, in SSPC we love these types of quad charts. So here we have the scientific challenges, so why we're doing what we're doing, why is it difficult, what's the payoff that we could get if we could actually do it. Then we boil that down into research goals, so tangible research goals. Here's our hypothesis that if we control the self-assembly of peptides, we can get more effective biopharmaceuticals. And the goal then to do that, like this is our dream and this is our operational plan, is to develop computer models to accurately predict peptide assemblies in a range of different pH, temperature, and strength excipients. So scan all those possibilities, all those myriad possibilities before ever actually going to the lab. Then use the simulation as a filter or a pre-screen, pick out the 10, whatever, but actually conditions and proteins which seem to be promising, and then test them experimentally. So always, yeah, I mean this isn't a one-way street. It's not like you do all the calculations, pass them to experiment, and you get the answer and everyone's happy. That it can happen that way. We'd love it did, but most of the time it's an iterative process. So we're helping to guide experiments, and experiments are helping us to refine and fine-tune our simulations. So it's a very, very stimulating environment to work in, and then when it works, it can work very well. So if it works, then the scientific impact is that we learn more about how peptides self-assemble, which is highly topical of interest for biomedical applications, for pharma applications, and then use those general design rules that we extract from simulations to understand structure-function relations from existing experimental data from our group and from collaborators outside of SSPC, and use that then to enable predictive modeling of morphology. So given a particular peptide sequence, what kind of polymorphs can we expect, and then use that to design new types of pharma materials. So in terms of impact on technology, if we can design peptides, well, they're structurally simple, they're uncomplicated, they're affordable to produce them on a large scale. It's really green chemistry. If we can discover their atomic scale assembly mechanisms, so figure out how they do what they do, that could enable rational design and precise control of peptide assemblies. So get away from trial and error experiments and do things in a very controllable way. Just like, I guess, inside our bodies, evolution has developed these highly precise mechanisms, these highly specialized environments in which to do the assembly. So we're trying to learn how to do that outside of cells. And yeah, if we get it right, then these peptide nanostructures can enable gene and drug delivery to specific sites in the body. So real smart pharmaceuticals. So then how do we do it? How do we use high performance computing? How do we use computational chemistry, computational physics to design better drugs using more complex APIs? So this was the first half of the talk, and this is all going to be the second half of the talk. So yeah, we want to develop a framework for understanding and predicting multi-component solid forms from first principles, so particularly co-crystals, and we also want to develop more complex APIs, so understand how to design peptides, how to understand how they assemble and crystallize. So basically we're a team of computational physicists and computational chemists developing new methods for molecular design, and the idea is that design rules that we obtain will be useful to actually make better API formulations. So one example is shown here. This is a dendrimer wrapped around a gold nanoparticle. So in terms of drug delivery, it's the ultimate in targeted delivery. One single molecule goes to one site in the body. Okay, so it's an attractive notion. We've designed these types of materials in the past. So how do we do it? We want to know what the structure is, what the dynamics are, and energetics. The structure is basically the PowerPoint science. What's the molecule like? How big is the particle? The dynamics is what we calculate. So we feed that in we build models, we put them into our computer, we watch how the atoms wiggle and jiggle over time in different solvents, in different conditions, and from that we extract energetics. So how favorable is it to form these types of single molecule structures? Is it more favorable under certain conditions for two molecules, for example, to bind at the same time to the nanoparticle? So can we do program design through figuring out the nanoscale interactions that occur? So here you've got chemisorption of these thioether groups onto the gold. You've got migration of gold atoms within the gold nanoparticle itself. You've got the molecule interacting internally, so you've got stabilization between these, say, different phenyl groups stick together, and then there at the same time you've got solvent, which I don't show here, but this is all done, say in this case, in dichloromethane. So you've got all the time a competition between wrapping of the molecule around the particle, or else the unbinding of the molecule back into solution. So the dielectric constant of the environment has a huge impact on whether or not these types of structures are stable. So why do we do this? Well, we'd like to make more effective APIs. So as chemists, as physicists, as biologists, knowing how these types of systems work is incredibly powerful for actually making new APIs and new ways of doing drug delivery. Of course, there's a value of death here. We all know about that horrible S-shaped curve between fundamental science and industry applications. I mean, a very, very small percentage of good ideas actually makes it into industry. We know that. The computers can help here, so we can try to avoid this happening, where good ideas turn to dust by using computers. So we can use high-performance computing to put numbers in nanoscale physics. So we can calculate and predict things like the binding energies between molecules, how they pack together in solution, how they absorb on surfaces, how they interact with solvent. We can do that on the computer before we actually go to the lab. So if we get the physics right, we can control the structure and the function at the nanoscale. And we know from statistical mechanics that if you get things right at the molecular level, then in a well-defined experiment and in a clean experiment, that should follow through to the experimental scale. So we do multi-scale simulations, we do quantum mechanics, classical mechanics, and everything we do is in partnership with experiment. So not just pretty pictures, everything we do is in partnership with experiment. So we're constantly refining our methods based against the sanity checks that we do, both within our own group and with external collaborators, to check that our methods are correct, so that these methods that we're developing truly are sophisticated enough and fast enough to feed into the development of better APIs. So yeah, in theory there's no difference between theory and practice, while in practice there is, which is clever and pretty, but it's also very true. So often things don't work. You know, we're lucky in Ireland. We have, as I say, I checked, the new K machine is very big. So we have, where we're batting above our average, I would say, as a country. We're lucky to have access to such a powerful computer. So that takes care of the first thing that hampers us in this type of rational design. So we do have access to bigger and faster computers. We could always use bigger ones. When iCheck isn't big enough, we go abroad. So in the past we've used computers, say IBM, have given us time on their massive computers in New York, and we also use computers throughout Europe when we need to. So when we have a sufficiently large scientific challenge that we can justify going to these super, in some cases almost exascale computers, we can do it. So we're not really limited by computing, to be fair. The big computers are there as long as we have a sufficiently compelling scientific case to use them. Something else we need, and that's part of the reason why we need bigger computers, is better statistical analysis. So if we're actually going to use our design rules for industry. So to actually make better APIs, not just new science, not just publications, but actually new IP, new ways of doing things, then we need statistics. We can't just be relying on a single calculation or a one-shot experiment. We have to really bulletproof our formulations. Part of that then is better linked with experiment, and also just better understanding of things like entropy and solubility. There's huge opportunities for clever mathematical physicists to actually contribute new knowledge on a very, very fundamental level, on a coding level. And in our group, we do a little bit of this, and it can really actually advance the field if you take the time to actually look under the hood of the codes and develop new maths essentially. So what do? We do modeling and design of complex materials at atomic scale resolution. We do it on big computers, and that allows us then to use quantum mechanics and classical molecular dynamics to treat large systems for long times. So we can make a tight and physically and in some cases even biologically realistic comparison with experiment. So what do? We develop things like nanoparticle drug carriers, membrane permeable drugs, and drug protein complexes, and peptide assemblies. So here you have the very difficult calculations, here you have the easy calculations. Within my group, this is a recent photo of our smiling faces, and there's around 10 of us. And we do both quantum mechanics and classical mechanics. And we also all invade each other's projects, and together it's very powerful actually. We were all good at different things, and we combine them, and then we can come up with new science stuff. So the quantum mechanics calculations are the hard calculations. Don't worry if you don't understand them, nobody does. Basically Feynman's idea was that you just believe the maths. Stop trying to frame everything in terms of your chemical intuition, because you're too big and too slow to actually understand what an electron does. But we have the maths to do it. So the answer is quantum mechanics. And the answer is this Schrödinger equation. That's it. Stop trying to make sense of it. We have that. We can use it to calculate very precisely the structure of small molecules. So for less than a few hundred atoms, we can use these very precise methods to calculate properties. They're the most realistic models you can get. They don't require any input from experiment. They're super clean, they're super elegant. However, to go to larger structures and to look at, say, intermolecular interactions in a very big, chaotic solvent environment in a large extended crystal, then we need to go beyond the few hundred atoms. So we need to develop potential energy functions based on quantum mechanics. So we take little molecules, we parameterize them using quantum mechanics, and then we generate these what we call force fields. So the most important part of the force fields are these non-bond interactions. So that's when two molecules meet each other, what do they decide to do? Do they run away from each other? Do they stick together? And that's described by this. This is the only equation I'm going to show you. Van der Waals interactions and Coulombic interactions. So Van der Waals interactions is just a balance between short-range electron-electron repulsion, long-range electron-nuclear attraction. It's the glue that holds the universe together. Very weak, it's very powerful, we have to have it in our calculations. And the other thing is electrostatic interactions. So that's, this from Leavenworth. Like charges repel each other, opposite charges attract. It's simple as that. So from quantum mechanics, we get the atomic charges, we get the precise amount of charge in each atom, and then we use this classical Coulombic electrostatic interaction to describe how strongly those interactions, how strong those charges interact with each other. And we include the solvent in everything that we do. So the solvent screens and in some cases mediates these electrostatic interactions. So with these classical simulations, we can treat millions of atoms for microseconds. So we capture an awful lot of the physics and the chemistry that's actually present in the experiments. We can get very close to the experiment by doing these types of simulations. But of course, for them to work properly, they have to be properly parameterized using quantum mechanics. So you have to get your quantum mechanics right, an individual molecule, with any hope of actually getting this part right. So there's a workflow there from quantum mechanics through the classical models. So we know from statistical mechanics that macroscopic properties that people measure in labs are often determined by molecular level behavior. But we also know that we need to benchmark everything against experiment. So we can get our qualitative information about macroscopic behavior from our atomistic calculations. We can in some cases even get quantitative information. But that depends on benchmarking. So high performance computing allows us to calculate the dynamics of millions of atoms over millions of time steps. Why would we bother? I mean, in iCheck we compete with MedAir to get computing time. So we have to have we have to need a compelling scientific argument for doing the calculations that we do. SSPC gives us that. So what do? We get the coordinates from crystallography or from NMR. We minimize the structure. That means we hit it with our force field, see what happens. That's a sanity check essentially. If the molecules blow apart, then we go back to the drawing board. We do our quantum mechanics again. Usually the molecule just sighs and relaxes a little bit. Then we assign our initial velocities from just a random distribution of numbers. Then we heat up to our temperature. It's generally room or body temperature, but in principle it can be any temperature we like. Then we equilibrate, so we allow the system to settle down. We allow solvent molecules to rearrange. We allow the solute molecules to form interfaces with each other. They'll do silly things in this phase because you're dumping in a lot of kinetic energy through heating. Strange things will happen, but over time the system will settle down. So this is like the teenage years. We don't really record that. By the time it settles down, by the time our energy flatlines, our temperature flatlines, then we're into production dynamics. So here we can actually, the dynamics is physically realistic. So we record the structures, and from that we extract energies which we use to predict the material's properties. Okay, that's the basic, in a nutshell, that's what we do. So I suppose the obvious question is, does it work? And the answer is yes, it does. Lots of the time it works. So the examples I'll talk about here, first of all, controlling protein mobilization on surfaces. So I'll just give you a very quick overview of just kind of snapshot of some of the stuff we do. This isn't by no way exhaustive, so it's just a very brief introduction. I'm very happy to share papers or share details with anybody who's interested. But for now, I'll just briefly throw some information at you, okay? So, for example, for controlling proteins on surfaces, it's two sides of the same coin. So in some cases, you want to try and direct the absorption of a protein on a surface, for example, to make lab on a chip applications, or maybe you want to absorb a protein in its functional orientation on a nanoparticle with its active site facing outwards. It's a very useful thing to be able to control the geometry by which a protein absorbs on the surface. And of course, the other side of it is that maybe if you want to store proteins in solution, you don't want them sticking, say, to the surface, sticking to surfaces. So whether you want it, whether you want to direct it or you want to prevent it, either way, knowing nanophysics, knowing the strengths of these interactions, these gluing interactions between the protein and the surface is very useful. So generally it's a competition between sticking of the protein to the surface and solvation of the protein. So what it wins by sticking to the surface, it loses in terms of solvation. So depending on how hydrophobic or hydrophilic that particular face, that binding face of the protein is, that determines whether or not it's going to happen. Okay, so we were involved for quite a few years with IBM, where we were designing organic films. So these monolayers of films on different substrates like metals, say, you can do it on plastics, you can do it on graphene, you can find them on just about anything. So just little thin organic layers. And we're very interested in seeing how proteins bind to those. So we used, a model protein again, this is a hydrophobin, and we found, okay, we get a mildly preferential absorption mode, so it binds most of the time through its hydrophobic phase, but it's very sensitive to environmental effects. So if you just change the chemical nature of the head groups of the film, if you change the salt concentration, you change the pH, you change the temperature, it's very easy to actually desorb these things. So we thought, okay, can we engineer stronger, more specific interfaces? So for something like this big monstrous, big ferritin protein, could we direct the specific absorption of that on surfaces? That would be a very, very useful thing if we could do it. So yeah, well, we worked with a very talented group of scientists at the University of Twente, where we tried to find, could we establish a molecular design protocol whereby we could actually force the orientation of proteins at surfaces. So take a typical protein, in this case it was just a simple barrel protein that's used every day in biochemistry, and see, could we force it to stand up or fall over? So like a little soldier, does it stay up or does it lie flat on the surface? And that's, if we can do that, then we can figure out maybe ways of engineering stronger, more specific interfaces, so we control the absorption of the protein on the surface. So this is Pascal and his wife Dorothy, who we worked with at NISA Plus Institute in Twente on this. It's a Science Foundation Ireland project. And this is the protein. So it's a tag RFP. It's a red fluorescent protein. It's a little barrel. And the idea that we came up with was to bind a hexahistidine tag onto it. So again, this is standard biochemistry. These are used every day all over the world in every biochemistry lab. So take this very simple prototypical system and see, can we engineer a surface that will bind specifically to it, or can we control the orientation of approach and binding of this very simple protein on the surface. So we had a gold substrate, we grew an organic film on it, we ended in acid groups, these NTA tricarboxylic acid groups, and we use this nickel tag to bind it to the surface. So this is not new chemistry by any means. But the clever part is that we're able, with their help, to design places to put the histidine tags. So we can put them here, 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 here. So we can design these different variants of the protein which have 1, 2, or 3 tags, and see, can we engineer whether it stands up or falls over. So from our molecular models, we can see if we've got 2 binders here, we can walk on the surface. That was very cool actually. We get this kinetically stable interaction between the 2 feet and the surface, and we can actually make it walk on the surface, which is kind of fun. So eventually 1 foot hops off, then as if this foot rises up, it takes long to come back, this guy comes off and you get desorption. So we're able to pull the protein all the way from a bound state to an unbound state. And from that, then we can use some physics to actually measure the binding energy of the protein on the surface. We measure the rupture force, basically how much force does it take to pull this thing off the surface, to rip it off the surface. And then we can just use reversal of times arrow to actually figure out just how strongly bound it was in the first place. Similarly, for the side-on orientation, we can do the same thing. So we rip it off the surface and measure the energy associated with that. We get these potential and mean force curves, and what they tell us when we integrate underneath these is that we get a strong preference for the side-on orientation. This guy wins. It's better to fall over. And that's because you've maximized the surface coverage between your protein and your surface. So all else been equal, it's better to maximize your contacts here. Generally, you'll get a layer of water in between anyway. So because it's a dynamic organic film, you get the best of both worlds. You get contacts of the hydrophobic parts with hydrophobic groups on the SAM, and then the hydrophilic parts can remain solvated. So you get a nicely balanced structure. What we find as well is that when you increase the coverage, they tend to stand up. So when you increase the concentration of protein, you increase the surface coverage, and then they actually nudge each other into standing up, because that way, then you also maximize the protein-protein interactions. There's a lot of different interfaces that you have to compute and be cognizant of in your design. But we had fun with that, and thankfully the experiments backed up our predictions. So what you see here are their confocal microscopy or fluorescence microscopy experimental results, and they show the same thing, that you get this change in orientation and surface coverage as you increase the local concentration of protein. So, and they did lots and lots more experiments with surface plasmon resonance, IR spectroscopy, etc. So we made a nice paper together on this. And the simulations were useful because they confirmed the multivalency. So because you've got more than one anchoring point, you get a kinetic effect as well. So as you increase the number of tags, you get an overall stronger interaction. So it becomes both thermodynamically and kinetically more stable on the surface. We also get some insight into the concentration dependence, where we see why we get a switching between side-on and end-on orientations. And also we get some details on that histidine nickel acid seal, which you can't get from experiments. So we see it's actually dynamic. Not all the 6 histidines are bound at any one time. So that also is kinetically stable. Because you've got 6 of them, if even 3 or 4 of them are bound at any one time, you've got a strong interaction. So you don't need all 6 to be bound all the time, which was something that was not at all obvious from the experiments. And that was a new insight that we got from the simulations. So yeah, on the atomic scale it looks something like this. So these are the nickels, these are the NTA, and this is the tag coming down. So you get this dynamic interplay whereby maybe 3 or 4 of the histidines is bound at any one time. Okay, so that's sticking proteins on surfaces. What about sticking proteins to each other? So we worked on this as part of a European project with the Weizmann Institute, a super talented, incredibly kind professor there by the name of Ed Beyer. And we worked a lot of things together, but one thing I want to tell you about is this Cohesion doctrine pair. So Melissa and Pierre in my group worked on this. Pierre is now working in SSPC. It's a very, very high affinity complex, but it becomes unstable at elevated temperatures. So these enzymes, this complex is part of an enzyme called the Cellulosome, which is involved in bacteria to break down recalcitrant sugars. It's a beautiful example of biological nanomachinery, but it doesn't work at high temperatures. So if you want to use this in bioprocessing, you're in trouble, because once you go to 100 degrees C or even 80 degrees C, this thing breaks apart. However, we found that in a thermophile, obviously as the name suggests, it does stay strong, whereas in mesophile it breaks at elevated temperatures. So that was the first thing Melissa did. She was able to confirm that depending on the sequence of the protein it's more or less stable at high temperatures. Then the clever part, which she developed, was to actually do a kind of a Frankenstein approach where you identify what part of the protein is important. In this case, it's this segment here, H2 and the cohesion. Just etch that off the thermophilic species, graft it onto the mesophilic species, and see do you get the resistance to temperature. And it works beautifully, actually. So these are the predicted binding energies. So this is where you mutate the mesophile, and it becomes far, far more stable at elevated temperatures. And that was confirmed experimentally at the Weizmann Institute, and again, it made for a nice paper. Then more recently, again, this is Pierre's work, working with Katrina O'Driscoll down in Cork. We looked at formation of complexes between, in this case, it's nucleotides and sugars. So again, biomolecules, soft organic molecules. In this case, it's RNA. In this case, siRNA. And these sugar molecules, these cyclodextrins. So we looked at a range of different complexes, one-to-one complexes, one-to-two complexes. And we also looked at the formation of superstructures of these functionalized cyclodextrins in solution. But then what we found is that they swarm around the nucleotide. So they form basically a little nano encapsulation particle around the siRNA. So you'll see this, I think, see here. Yeah. So what Pierre and also Rakav Singh, who worked on this study, and what they did is they gradually ramped up the concentration. Here you've actually got 39 cyclodextrins and 1 siRNA. So you can see how it's completely smothered by the sugar molecules. So because they're functionalized in these cationic groups, they form these very stable electrostatic interactions with the negatively charged siRNA. So you're basically making a self-assembling drug delivery vehicle for the siRNA in water. And the guys in Cork did some calorimetry experiments which backed up our conclusions on this. So again, it's a nice example, I think, of predictive modeling of something that's, I would say, impossible to see using experiments alone. So what we see is that these siRNA, they're naturally negatively charged. These molecules serve as an ideal template around which the cation functionalized cyclodextrins rapidly assemble into stable multimers. So it's like they kind of nucleate around nucleotide. So that makes a promising, stable gene delivery vector, because these things can protect siRNA that is actually injected into cells. So you get essentially a lipid-like behavior of those cationic cyclodextrins in solution, and you get this capacity to form stable superstructures. So they essentially provide nanoscale molecular templates with highly controllable shape and size complexity. So it's true molecular and nanoscale engineering. The amphiphilic nature of the complex, so it's got cyclodextrin here, so that's hydrophilic on the outside, hydrophobic on the inside. And then you've got here a very charged region, so it's amphiphilic. And the resulting bilayer formation that comes from that, it's very much biomimetics. It's copying phospholipid bilayers. They can aid membrane permeability. Because you've got that bifunctionality, these things should actually be swallowed by cells. So we're currently testing that. Well, guys in Cork are testing that experimentally to see does that enhance the transfection activity. Okay, so I'm almost done. I just want to leave you with some ideas that we're currently developing on designing peptide nanostructures. As I say, we got started on this through a European project, again, at Melissa and Pierre's work, where they looked at the Cellulosome enzymes. This is an example of a designer Cellulosome biocatalyst that they developed. So it's got lots of different protein units all strung together by these dynamic linker molecules. So you form this beautiful multifunctional nanomachine, essentially. And this is very, very good at eating cellulose. So we spent quite a lot of time on that. And we developed new methods. This forced us to develop accelerated sampling methods, because you simply have to, you can't just like stick this into your simulation and expect it to change significantly over time. It's just too big. So we had to develop smart ways of actually dancing around the free energy landscape for these types of very complex systems. And we're exploiting those methods that we developed now in SSPC to look at peptides. Also, Osen, PhD student in my group, Sarah, who's now a postdoc in SSPC, have been looking at dipole-directed packing in amino acid crystals and also assemblies. So Sarah does the quantum mechanical calculations. She calculates the dipoles, and from that predicts the type of structures that you form. She's currently working also on co-crystals, using the same design principles that she developed in this work. And Osen then tackles the problem from the point of view of classical molecular dynamics. So he makes basically super cells of these types of peptide crystals, then dumps them into a box of water and sees how long it takes water to actually etch in and fray the crystal. So he's developing methods to do that, essentially to measure the solubility of these types of complex crystals. Just to give you an example of kind of type of workload that we use, this is developed by Sarah. So first of all, you do the atomistic prediction. of things like the elastic properties, dielectric properties, the overall formation energy. Then from that you select the most promising candidates. So whether they're, say, crystals, co-crystals, films, whatever it is, the simulation allows you to get a first computational scan of the surface and predict the most useful ones. From that, then we do validation of the predicted physical constants. So once we have the material, we can then either in-house or through collaborators actually validate those predicted physical constants. So with piezoelectricity, we can do it ourselves. For elastic constants, we can do most of it ourselves. And then for, say, on the drug activity, then we go to collaborators for that within SSPC. From that, then we design possible tests. Say for piezoelectricity, it's energy harvesting test. For SSPC, it would be tests of drug efficacy based on the simulations. And then because we're working directly in partnership with the experimentalists, we help each other. So when this works well, we get better at doing the calculations, the experimentalists get better at doing the experiments, and together we develop nicely materials. I'll leave you with this. This is work being done by Sean Bhattacharya, who's now a collaborator in SSPC, Jeffrey, who was a PhD student in the group, he's now gone to industry, and Liang Zhu, who was a postdoc in the group and has now gone back to America. We worked with a group in EMPA in Switzerland to look at controlled growth of peptide oligomers and fibrils. So these are peptides that are implicated in Alzheimer's disease, so amyloid beta 40 and amyloid beta 42. So we managed, using a combination of simulations and experiments, to actually image those oligomers. So one is shown here, for example. It's a low molecular weight assembly of these proteins with sub 2 nanometer resolution. So we can do that without the need for fluorescent labeling or metal staining. So we really capture them in a native state. So by combining qualitative and quantitative data that we obtained from this work using simulations and experiments, we get new insights that could contribute to further structural determination and to investigate their cytotoxic nature. Because it's well known that some oligomers are not implicated in cell death. So we think we managed to actually identify the important oligomers. That's always the crux in this field, is to actually figure out which particular nanostructures are actually doing the damage. So this is what our friends in EMPA see with their microscope. So they just see these little dots. Now they only see them for amyloid beta 42 and they don't see them for amyloid beta 40. So that was a nice sanity check on the experiments, because it's well known that AB42 is the more toxic one. So, okay, they see these little bright spots which we think could be low molecular weight oligomers. So that's where the modeling comes in. These are wonderful people, we love working with them, but this is as much as they can see. So that's where you really do need to be coupled with computer simulations to actually resolve the structures. That's how so that's from the simulations then, from the hard work by Xian and Liang and Jeff, we managed to generate molecular structures. So we compared the height profiles that we get for different sized assemblies with what they measure using microscopy. So they measure basically width and the height of those little red dots. And we can then fill in the atomic details from our computer models that match that. That's our guide. The heights from AFM are our guide. So we can see lots of different poses that they take depending on whether it's amyloid beta 40 or amyloid beta 42. We calculate the conformational energy, so that's basically the stability of these in different folds. And what we find is that we get key differences in stability and tendency to form fibrils. So the orientation in which the molecules sit determines their propensity to form fibrils. So we can explain the experimental data based on some very subtle asymmetries that we see in the simulations. So that's basically a whirlwind tour of what we do, and I'm happy to answer any questions you might have. So this work was done obviously with the help of a lot of people, group members past and present, and I know I speak for all of my group, also all of the SSPC modeling group, particularly the atomic scale modeling group, when I say that we're super excited about the possibilities of actually employing these tools that we've been developing for the last few years to joint, co-discovery projects with experiments. So using combination of experiments together with simulations to design new pharma materials. Thank you very much for listening.